Here we will be discussing about vegetative propagation. The main objective of this session is to know vegetative propagation and the different types of vegetative propagation, artificial vegetative propagation, and what are the advantages and disadvantages of vegetative propagation. We know that all the plants in the world are unable to produce seeds, but some kind of propagation methods are there in those kinds of plants. Here, vegetative propagation is an asexual method of plant reproduction that occurs in its leaf, roots, and stem. This can occur through fragmentation and regeneration of specific vegetative parts of the plant. This occurs when plants grow and develop naturally without any human interference. We know that there is no role for human being in the case of natural vegetative propagation. Here, we will be discussing about the different kinds of vegetative propagations. The first, going with vegetative propagation through roots. So from this picture, you can see that there are a lot of tubers are present in the sweet potato. And when we are planting these tubers, they will be able to grow as a new plant. So the best example for propagation through roots are carrots, sweet potato, and dahlia. And moving to the next, propagation through leaves. From this picture, you can see a lot of leaflets or plantlets are there at this margin of this leaf. Once these plantlets are matured, they will be falling down from the parent leaf. And if the conditions are favorable to that plantlets, they will be growing as a new plant. The best example for this category is bryophyllum and begonia. And moving to the next, that is vegetative propagation through stem. You can see from this picture, we can cut the part of the stem and plant it in a pot or in the ground, and this stem part or this piece will be growing as a new plant. This is what I mean by the vegetative propagation through stem. The example for this category is rose, sugarcane, hibiscus, and potato. And now we will be discussing about how can we do some artificial vegetative propagation. Till we have discussed about the natural method of vegetative propagation, there is no involvement of human being in that situations. But now we will be checking how can we make some propagation in the case of plants. This is a type of vegetative reproduction carried out by humans on the field and in the laboratories. The different kinds of artificial propagation are cutting, layering, grafting, and tissue culture. From this picture, it is very clear that we can cut the part of the plant. It can be a part of the stem and plant it in some other place and provide all the nourishment, water, etc. And that piece will be growing as a new plant. Examples: mango, lychee, lemon, rose, etc. And the next, layering. The stem of the plant is bent down until it touches the soil. The stem is then cut once it develops roots and grows into a new plant. So when the stem part is touching in the ground, if the soil is moist and all, of course we know that there is a chance of growing roots in that part. Once the roots are developed in the covered part, we will be cutting both ends of the stem and that plant part is able to grow as a new plant. Layering are of two types are there. I will be showing the different kinds of layering in the next slide. Next is grafting. The stem of a plant is cut and then fitted on another strong plant and covered with some grafting wax. From this picture, you can see that there is a strong parent plant is there, that is known as stalk, and a new stem piece is there, that is the desired plant piece, and that is known as scion. And we will be cutting both the ends of the plant stems in a desired way to fix them together. And we will be fitting them together and applying some grafting wax on the surface to restrict the entry of any kind of moisture. And we will be covering or tightening with some cotton threads or any kind of plastic bags and all. And the last, that is tissue culture. It is a collection of techniques used to maintain or grow plant cells, tissues, or organs under sterile conditions on a nutrient culture medium of non-composition. Here I will be going to discuss about the layering and grafting and tissue culture in little more detailed way. This picture is showing the different kinds of grafting techniques. You can see bud grafting, that means the desired bud from another plant we will be taking and we will be trying to fit it in the stalk, that is the strong parent plant. And like before, we will be applying some grafting wax and all and tying it properly in the parent plant, so it will be growing as a new plant later. And the second one is showing that approach grafting. You can see the stem of two plants are tied together, right? In this way, what are the desired plant part we are taking? We are able to fit them into the stalk and give all the precautions for that plant to grow properly. These are the methods of different types of grafting. And moving to the next type, that is layering. Here, two types of layering techniques are showing, that is mound layering and air layering. In the mound layering, you can see that the branch part is about to touch or touching the ground, and if the ground is wet enough, of course we know that the plant parts are touching in the ground, there is a chance of growing the roots over there, right? Once the roots are formed in that part where the stem and the ground is in contact, we will be cutting the other end, and that part is able to grow as a new plant. And in air layering, we are taking a desirable branch and we will be removing the bark from the stem piece. We are taking a handful of the moist soil and we are fitting or we are pasting that moist soil in that part where we have removed the bark of the plant. And after that, we were taking some polythene bags or any pieces of the polythene and tying, and we want to avoid the presence of the moisture over there. Okay, in all the situation, we are applying the wax and the polythene bag for controlling the presence of moisture. This tissue culture. Here we will be taking the desired plant part and transferring this plant part into an agar medium. Agar medium is a medium which provides all the nutrients for the plant for the proper growth. And other than that, it will be having some auxins which will be boosting the growth of the plants. And after keeping this plant parts in this medium for some days, they will be developing tiny plantlets. After getting into certain growth, we will be planting this into different compost and we will be taking care of after. Okay, this is the method of tissue culture, and this is showing some pictures of tissue culture techniques. And after that, we will be discussing about the advantages of vegetative propagation. Vegetative propagation is faster and more certain method of propagation. New individuals produced have exactly identical qualities as their parents. They will be maintaining all the good qualities of their parents and can be used to produce plants that do not have viable seeds. That we have discussed already in the beginning of the session. The plants who all are unable to produce the seeds can propagate through their vegetative propagation through root, stem or leaves. Certain desirable characteristics present in the plant can be maintained. So the good qualities of the parent plant can be maintained in their next generation. And now we will be discussing the disadvantages of vegetative propagation. New plants are less varied. It's a type of asexual reproduction. There is no mixing of two genders, no mixing of different genes. As a result, they will be identical to their parent plant. So variations are very, very less. And when we consider the cost, they require skilled labor and aftercare. We have to take care of the plants. In the case of the tissue culture, grafting, layering and all, we know that we have to take care of the plantlets or the small plants. And it is little time consuming. Each plant has to be individually propagated. And lack of dispersal mechanism may lead to overcrowding. So there is no method of dispersal by wind, water or any other agents. So there is a chance of overcrowding of the small plantlets. New plants may be less adaptable to the changes in the environmental condition. This point is related with the variation. If we can't see much variations in the kind of vegetative propagation, so there is a lot of chances of less adaptive features in these plantlets or the new plants. Susceptible to diseases. This is also the same thing. There is no variation, so the adaptation capacity will be less. At the same time, they will be prone to different kinds of diseases also. These are the disadvantages of vegetative propagation. So in this session, we have covered what is a vegetative propagation, what are the different methods of vegetative propagation, and what is artificial vegetative propagation, and also what are the different types of artificial vegetative propagation. And as last, we were discussed about the advantages and disadvantages of vegetative propagation. Thank you.